The next process we're going to be talking about is translation. And translation basically has to do with taking your messenger RNA that you just made during transcription and reading the genetic code to translate it into a protein language. So this is an example of the genetic code, this table 5.3, and it shows you all the amino acids that are specified by the codons of the messenger RNA. This is posted up on Laulima under resources, so please make sure you print yourself a copy of this because it's going to be very important for any quiz that you have in this section, as well as exam number 3, which is going to be covering a lot of the genetics topics. So if you notice on this, there are 64 possibilities. Again, every particular one of them is a 3-base code. So if you look at some of the ones you have here, you have AUG, which is methionine. Every protein always starts with AUG. So this is the messenger RNA that I just took from the last process. And again, this is translation. And translation is when you take your messenger RNA and you read it into a protein language. You're going to be looking at the particular amino acids. And so let's look at what we've just coded on that small piece of DNA that we just took. So here's your messenger RNA. And your first code is AUG. So looking at your chart here, you'll notice AUG stands for methionine. Methionine is always the first amino acid of every protein that you make. Then you look at CGG. CGG on here, find it, is arginine. That would be your next amino acid in your sequence. CAU is histidine. And lastly, we have UGA. And if you look at UGA, you'll notice that it says end chain. So this is an end chain or something called a stop. Every protein starts with methionine, which sometimes you'll see it as the word start, and every other one ends with an end codon, which is always stop. So where does this process take place? We know that proteins are made in what organelles? In the ribosomes. And so when this particular message comes in contact with a ribosome, it simply rolls through the ribosome. So if you imagine the ribosome having two subunits, it has a top and a bottom, a 30S and a 50S subunit, your messenger RNA is going to roll through here. So here's our AUG that we just looked at on this side that we just coded. AUG, CGG, CAU, and UGA. So what happens is the messenger RNA starts to come in contact with the ribosomes, and the ribosomes clamp down on it, and they start to roll. So the ribosomes will be moving in this direction, the message is going this way. When it comes in contact with the ribosomes, a particular structure called a tRNA, which looks like an upside down T, rolls into the ribosomes, and it has a particular amino acid on the back of this that it brings in. So in this case, the first one that's going to come in is methionine. It's going to have an anticodon region that matches up to AUG. There is only a place for 2 tRNAs inside the ribosome at a time, and so as this is rolling down, the first 2 will be placed in there. The amino acids will link together as we showed you here, and when it reaches the last one, which is the stop, the ribosomes will kick the tRNAs out of place. The 2 ribosomes will be separated and wait for the next sequence of messenger RNA to come into place. So if you're looking at the processes, again, replication takes place in the nucleus. Transcription starts in the nucleus where you make your messenger RNA. Messenger RNA then goes out of the nuclear pores to the cytoplasm to find the ribosome, and then translation occurs on the ribosome. The ribosomes clamp down on the messenger RNA itself. The messenger RNA rolls through, and the new proteins are being produced on the ribosomes, and then once it reaches a stop codon, the entire intact protein is then dislodged, and then you have a functional protein. So those are the 3 processes, again, where they take place and the end results. Again, with this particular strand, you have methionine, arginine, histidine, and an end codon or a stop for this particular small protein that we made. The smallest proteins in nature, the beta globins, is about 120 amino acids, and some of the largest ones are about 120 to 160,000 amino acids, and this is your gamma globulins, and also egg albumin. They run at about 120,000 to 160,000 amino acids. They're incredibly large amino acids, incredibly large proteins. Okay, that's translation.